Okay, so wrapping up chapter 4, remember you have an organelle quiz tomorrow. I posted the organelles onto Google Classroom. Those are the organelles. There's a table in your book that lists all the organelles, and I think it says structure and function. So use that, make your own little notes. But wrapping up chapter 4, looking at cytoskeleton and extracellular structures. So the cytoskeleton is basically a bunch of fibers that shape the cell, support the cell, and also can help the cell move. You may not know this or think of it, but your say cells, they have these fibers, they act like crossbeams throughout the entire cytoplasm, and they actually help anchor organelles and can move organelles. So like during the process of cell division, after the mitochondria has divided into two, has copied itself, it'll hop on one of these fibers, and it's like a railroad track leading it to the opposite ends for cell division. So we're going to talk about how organelles or even vacuoles and vesicles can move using the cytoskeleton. This system is very dynamic. It's always changing. The cells do change shapes. Like they can move. We'll talk about different cell movements, but these fibers, they will detach and move and reassemble every second that it's alive. So there are 3 types of fibers that compose the cytoskeleton. So I'm going to talk about each 3 and break it down. So the first one is the actin filaments. I'm just going to abbreviate AF. These guys, they are long fibers, about 7 nanometers, composed of 2 protein chains intertwined together, as you can see down here. You can kind of think of it as 2 pearl necklaces where the beads are your actin proteins. They do have polarity, which means it has a positive end and a negative end to designate direction of growth. And it's also, its main function is for cellular movement. So cells that can contract, cells that can crawl or pinch, actin filaments are being used there. And so in this process of the positive end, the polarity, the plus end, and the minus end, well, the minus end means that it will depolymerize, meaning we'll take away a bead and we'll move it over here. So the positive end is the growing end, and the minus end is where it breaks down. It does take expenditure of energy to do this. As you can see, we're using some ADP, converting it into ADP to depolymerize and repolymerize at the positive end. Second type of fiber in the cytoskeleton is the microtubule, or MTs. These guys are the largest. They're just huge hollow tubes made of some beta protein filaments. They form at nucleation centers near the cell center, and then they radiate out. So I think I might have a diagram of this on the next slide of what it looks like and how it kind of radiates out. But it's always in a constant state of flux, which means that it polymerizes and depolymerizes, which means it elongates or breaks down. It can last up to 10 minutes to 20 seconds. But in this case, the positive end, so if this is my cell, and let's just say this is like the center of the cell, and we have microtubules that extend out. The positive ends grow away. So this is positive, this is negative. So negatives towards the nucleation center of it. Does provide some organization to the cytoplasm, and I like to think of microtubules as railroad tracks. They are responsible for moving materials within the cell itself. So organelles, if they need to move to one end, to prepare for cell division, they will hop on a microtubule and travel along to its destination. So here's a microtubule with the beta protein subunits. It is hollow on the inside. This diagram is showing the negative end and the positive end for rapid growth. It can contrast. It can be very dynamic. It can happen very quickly, 20 seconds, 10 minutes. We use microtubules as well during the process of mitosis and meiosis, moving those chromosomes to the center and then pulling them apart. Okay.
Let's have the door open. Big table saw. Not even shutting her off.
All right, and then the third type of fiber found in the cytoskeleton are the intermediate filaments. So they're medium sized. So they're not as large as microtubules. They're not as small as the actin filaments. They're in the middle. These guys, most durable, very tough fibers, protein molecules intertwined together. You can see that it is larger than the actin filaments. These guys are stable. They don't break down. They hold organelles in place. I love this side view of it, just showing how they just intertwine and give support. Organelles, they can just kind of like rest, in this little nook, kind of like a hammock, until it needs to be moved again. So intermediate, you find these guys, keratin, main ingredient keratin, vermentin. I don't really know what vermentin is, but I know what keratin is. It's your epithelial. Neurofilaments, so they're also found in your neurons. Okay, so this diagram is showing how all three kind of work. So here we have our intermediate filaments, and they're just cross bridges all over, and you can see that they're holding organelles in place. We have our actin filaments. They're usually found towards the outside of a cell, and then the microtubules acting like railroad tracks, transportation system. So this shows the intermediate. There's your microtubules, and then the actin giving support on the outer regions of the cell. So with microtubules, we have to talk a little bit about centrosomes, because centrosomes, basically they just have microtubules radiating out of them. So centrosomes, they're basically centrioles, barrel-shaped organelles. A lot of times in diagrams you see something like this at 45-degree angles. Maybe when you guys made your cell models, did you make centrioles? Anyone here? Okay, some people did, because you don't find centrioles in plant cells. If you did a plant cell model, you should not have made a centriole in your cell. Probably lost points if you did. But they are usually depicted as 45-degree angles. So yes, paired, located at right angles to each other. And then the centrosome is just the region that surrounds that pair. So centriole, centrosome, basically. But from these centrioles, we have microtubules radiating out, and then they hold chromosomes and pull them and move them to where they need to be. So the diagram, way better diagram than mine. 45-degree angles, there's the microtubules.
I don't know, I like yours better.
Thanks. A lot of times you'll see a pericentrial material, kind of like a fuzziness around it. So if we go back, kind of this fuzziness around that, that's your pericentrial material. So it's just a ring-shaped that surrounds kind of everything, and it's kind of like the building blocks for all the microtubules, those subunits that depolymerize and repolymerize. Remember, you don't find centrioles in plants or fungi, but they do have microtubules inside of them, just not centrioles. So with cell division, we use the cytoskeleton a lot. Actin filaments and microtubules are at work. So when chromosomes are moving within cell division, you're using microtubules. Actin filaments are used to pinch that cell in half. So in animal cells, we get the cleavage furrow going on here, where it's just, basically a ring is formed around here. I know this is a 2D picture, but the actin literally forms, it's like a belt around the tube, and then all they do is tighten it. So then these actin will fall off, and then they'll reattach and just make the ring smaller and smaller and smaller until you have pinched the cell into two. Muscle cells also use actin filaments to contract. So in second semester, we'll talk about human body and how muscle cells attract. So actins, you'll revisit that again. But both provide scaffold for cell shape and movement. They can hold certain enzymes in places within the cytoplasm. Okay, so now I'm going to switch gears and talk about how organelles and things can move within the cell. So one way is that you can use the endoplasmic reticulum. We talked about vesicles, the endomembrane system. A protein is made, it gets packaged in the ER, sent to the Golgi apparatus, and then from there can become maybe a lysosome, can leave the cell, or go to its destined location. So we can, say that this is the intracellular highway. You move these vesicles They're loaded with cargo, but it's going to be used in the cytoskeleton like a railroad track. So what's moving the package or the cargo, if you will? Well, you kind of need these molecular motors. So down here we have kinesin and dynin, and these are our molecular motors that move organelles or vesicles along the microtubule. So the vesicle organelle has to be transported. That's your cargo. You have a motor protein. It does need to use some energy, so a lot of times ATP or another form of energy is involved. You need a connector molecule that connects the vesicle to the motor, okay? And then finally you need your railroad track, which is the microtubule. So the direction depends on the motor being used as it's traveling down the positive or negative end of the microtubule. So there are two kind of connector molecules, okay? You have connectin and dinactin. So these are the connector molecules, but the motor protein, well, connectin connects the cargo to kinesin. With me so far? Okay, yeah, okay, dinactin, which is the connector, connects the cargo to the motor protein dynin. Okay. Now, if you want to go away from the center of the cell, like outwards, so remember, this is the center of the cell, and we have microtubule as the positive end, the negative end. If you want to leave or go towards the outside of the cell, you're going to use the motor protein kinesin. So kinesin only moves towards the positive end. Dynin, this is supposed to be a minus end. I don't know why that went down there. Goes the opposite direction towards the center of the cell. So don't get those two mixed up. Guaranteed multiple choice question on the exam, talking about either kinesin or dynin, or even the connector protein or molecule itself. So, okay. Well, I usually show a video after this. I'm going to show you a video of how connectin actually works. It's kind of cool. So I'm going to go back to this diagram. Okay, so here we have the package, okay, the connector molecule. This is the motor protein down here, and they kind of look like little legs, little Smurf legs almost, right? Actually, how it works is ATP will bind to this, let's say this leg. We're moving this way. Will bind to this leg. It will detach and literally swing over and grab the microtubule and latch on. Then another ATP molecule will come and latch onto this leg. And so it literally walks hand over hand down the microtubule. Or in this case, the legs, they do something really creepy. It's like a toy leg where you can like spin it all the way around. So it's like, yeah, it's crazy. So they actually have captured this on film of the motor proteins walking along the microtubule, and you can see the legs just kind of swing and yeah. Okay. So in this, they somehow attached beads to kinesin. Okay, so we're gonna this is the center of the cell, and we're gonna march this way, I believe. And they just kind of crawl along. Okay, there's another one that's way better. Hold on, I just haven't found it yet. A day in the life of a motor protein? That looks like an interesting video. Oh boy, it's 5 minutes long. You know what? The first 15 seconds, nope, 5 seconds is what I want. Okay, this is a computer diagram of the motor protein moving here. So, watch the feet.
brain.
Yeah, so I think those were feet. I don't know.
The motor protein kinase.
Hold on. No. There's one more where you can really, in real life, see the legs swing. It's kind of creepy looking. No. Okay, I'll just show this one. I'll just show a little bit of this one, and then we'll just move on. Yeah, but you get the point, right?
Oh, yeah.
Sort of, hopefully. Okay.
Wait, what are those things called?
That was kinesin. Mm-hmm.
Oh,
Wait,
versus
so like,
the dyne.
why are they walking? Where are they going?
Well,
They're
like I
just
said,
killing.
maybe there's cell division, or maybe this vesicle needs to leave the cell, so it's gonna.
Oh, so they just do like whatever.
It just moves things with inside the cell. That's how
Think
things
about
move.
being a little cell. You never get to have any fun time. You just feel like starting
Always work.
Literally.
All work, no.
And if you stop, you just die.
Think about being a little cell. No fun. I do. I usually do. That's all I think about my life,
Okay.
Always
So cell motion is tied to actin filaments, microtubules, or maybe they use both. Our intermediates, they're kind of like tendons. They just prevent from excessive stretching because if the cell were to overstretch itself, it may, tear the plasma membrane. So there's a limit as to how far a cell can stretch before it's in trouble. Actin filaments, major role in determining shape, and it can do this very quickly as to what shape the cell will take. So here we have a cell and it's kind of crawling along. And so we have the actin that stretch out, they depolymerize and repolymerize, okay, and they kind of extend the body out, and then they'll form an attachment to the surface or whatever fluid that they're in. And they'll attach, and then they kind of depolymerize on this side and move stuff. And it kind of rolls or crawls along.
It's like a slime.
Yeah. Some cells crawl by the arrangement of these actin filaments. Huge significant cellular phenomenon. I mean, this is how your white blood cells, move during inflammatory as they're, squeezing its way through the capillaries to the site that's injured. Clotting for wound healing. This is usually how cancer spreads. On the leading edge of the cell, our actin filaments rapidly polymerize, extend out forward, then they stabilize, and then once they're stabilized, the back end will, detach and kind of just roll or even slide along in the direction that it needs to go. Now you're probably asking, how do cells know where to go? A lot of times on the plasma membrane, there's receptors, or maybe something that can detect food if it's a unicellular organism that will tell, that directs the movement of the cell. So again, another diagram showing it as it crawls along the surface. It attaches, and then we contract on the opposite end, and then extend further out in the direction we want to go. Cells can also move by using flagella and cilia. So eukaryotes and prokaryotes have different kinds of flagellum. This is really important. Eukaryotes, their flagellum is arranged in 9 microtubule pairs surrounded by 2 central microtubules, or a 9+2 structure. So this is what I mean by a 9+2 structure. Here are my 2 center microtubules, and then if I count on the outside, we'll start here. 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 pairs. So this is a 9-2 structure for eukaryotic cells. So if you were just given a flagellum cross-section and you're like, tell me if it's eukaryotic or prokaryotic, you're looking for a 9-2 arrangement. If you see a different type of arrangement, you say it's prokaryotic. Another difference is that the eukaryotic flagella undulates. It doesn't rotate like a propeller. Instead, it's kind of doing this. Think of like a snake motion, okay? Flagella is also an outward projection of the plasma membrane, so keep that in mind. But the microtubules are derived from something called the basal body. So in this diagram right here, here's the basal body. The microtubules extend out and become part of the flagella. Cilia are a little bit different from flagella. Shorter cellular projections, and you usually see them in rows. So down here, looking at this unicellular creature, you can kind of see that it looks fuzzy and hairy. Well, that's all cilia, and they kind of act like little rows, if you will, to swim or stroke across the medium that they're living in. So usually more numerous on the cell surface, but same internal structure, just different functions. We have cilia on our epithelia for nasal passages and throat, and like mucus kind of collects on the cilia, and traps dust and bacteria from getting into your lungs. Okay, so yeah, I'm not talking about like nose hairs. I'm talking about microscopic. Okay. Plant cell walls also provide protection and support. So we talked a little bit about the plant cell wall, how it's made of a polysaccharide. What's the polysaccharide?
It's a sugar and multiple sugars.
Multiple sugars linked together, and in this case with plants, it is what?
Oh.
Cellulose. Okay. Fungi cell wall is composed of chitin, which is also a polysaccharide. But there are some differences between the cell walls. Fungal cells do not have chloroplast, plants do. Fungal cells are heterotrophic, plants are autotrophic. So heterotrophic means they eat other things. Yep, so they usually secrete digestive juices and then reslurp them back up in some way. Plants make their own food using sunlight. Fungal cells have rigid cell walls made from chitin. Plants is cellulose. So with plants, we have primary walls that are laid down during the cell growth when it grows most quickly. And then in between, we have something called middle lamina that glues the cell together. So where am I here? Primary, okay, secondary, and then middle lamina that glues it to other cells. Some cells produce a secondary cell wall. So, yeah. Animal cells, instead of a cell wall, we tend to secrete something called an extracellular matrix, which I will abbreviate ECM. It is an elaborate mixture of glycoproteins, so that's sugar and proteins in the space outside the plasma membrane. We can also have some fibrous protein called collagen interwoven in there, or maybe some fibrous protein, or even elastins. So it's like a mini cytoskeleton outside of the cell. Okay, but we call it the extracellular matrix. Attaches to the plasma membrane by a third kind of glycoprotein called the fibronectin. You may need to know what the fibronectin is. So I have a diagram on the next slide that shows you what the ECM looks like and then how it's connected to the plasma membrane. So here's my collagen fiber just kind of woven outside of the cell. Here's the phospholipid bilayers. We got some protein embedded into it. And then right here, these little green guys, green tubes, whatever. That's your fibronectin that's connected in the ECM to the actual cell. And then they break down the ECM right here, and they show you, okay, we got some carbs bonded to some proteins. Here's a big polysaccharide fiber. So it's a very complex structure outside. And the purpose of it is to maybe latch on to other cells, maybe to help communicate with other cells. So fibronectin binds to the ECM glycoproteins, and those proteins are called the integrins. So if I go back, this is the integrin, the proteins that are embedded into the plasma membrane that help the ECM attach to the cell. Lots of vocab. Integrins extend into the cytoplasm and attach to microtubules and intermediate filaments. And why does it do this? Well, it can help influence cell behavior depending on what cell type you're looking at. It can also alter gene expression. Sometimes cells go into like an arrested state during cell division. Hey, we don't need you to divide, don't copy your DNA, or maybe don't make this protein. And then also it helps during cell migration. So I think in chapter 19, later on this year, we'll talk about how cells migrate in the embryo and how you form a head and a tail, and we're going to come back to integrins during that. So. quiz tomorrow. Please, 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 please study. Did anyone find the table in the book that I talked about? Okay. Alright.
Oh, wait. That table
Can I
in the book,
finish this?
In chapter
No,
4.
I lost it. Oh yeah. I'm pretty sure it's in my book at home, but if not, looks like I'll have to. Do you have this part
Good
on?
either.
No. That's the one for. Yeah, me too. I
That-
haven't passed on.
Yeah, so for those watching the lecture, it's page 79 in your college biology book, table 4-2.
Have it labeled.
What do you mean?